it is time to move on to the next presentation. It's a new presentation for the course, it's from Katherine Siddle from Harvard and the Broad Institute. So thank you very much for joining us to talk about a new technique that was recently published. I think it was in Nature Microbiology, I can't remember, but I'm sure you'll talk about it, about how you can try and track problems that can occur in the lab using DNA spike-ins. So I'll hand over to you. Thank you very much.
Thank you. Yeah, nice to be joining this, this workshop. I'm going to try to share my screen. How does that look?
Looking good, yep.
Okay, fantastic. All right, so yes, and I think as was noted, I think this has been, you know, fortunately, my talk has been really well set up by the previous discussion and by everything Nabil has already introduced. So as Nick said, I'm Katie, I'm a postdoc at the Broad Institute and Harvard University and the lab of Paddy Sabetti in Boston, Massachusetts. Yeah, and I wanted to talk about this. I'm mainly going to focus on this one particular method that we've set up for detecting and tracking contamination. But I think by way of doing this, the point that I really want to emphasize is that as you get into ARTIC sequencing and multiplex amplicon sequencing in general, it's really important to get ahead of contamination, and that contamination will happen. It does happen. You know, we need to put it in multiple mitigation steps. And I think Nabil already alluded to several of the ones I'm going to talk about, and also how you can pull this out at the informatics stage as well. But it's, you know, we need to put as many controls in there as we can to pull out contamination and pick it up where it happens. And in particular, we found these in-well spike-in controls to be a really powerful way to do that. So just by way of an introduction and background, I wanted to kind of explain sort of how we got into this and why this contamination thing was something that was top of our minds. So the lab that I work in actually has been doing viral genomics and genomic epidemiology for many years on a lot of different viruses, mainly sort of Lassa, Ebola, hemorrhagic fever viruses, before we got into, as everybody else, in the spring of 2020, SARS-CoV-2. For the first wave in sort of the spring of 2020, we actually were using primarily metagenomic or unbiased sequencing methods in order to sequence SARS-CoV-2, but also sort of detect other pathogens. And this was an approach that we had available to us in the lab and that we were, you know, comfortable using. But we realized through this first wave that really, if we were going to meet the scale that was needed of sequencing, we were going to have to switch to a multiplex amplicon approach to get both the sensitivity for high CT samples and also just the throughput and speed that was going to be needed. But we were, because of our background in metagenomic sequencing, we were actually, you know, a little bit anxious about the massive amounts of amplification that was done by these multiplex amplicon approaches, and wanted to make sure we were kind of getting ahead of that risk and had something in place before we started in order to detect contamination. So we spent this sort of fall of 2020 really thinking about this deeply, and then switched to amplicon sequencing with this sort of winter 2020 wave. And the approach that we settled on was that we felt really strongly that we were going to need more than just water controls on a plate, but we were going to need something in every sample, in every well, if we were going to pick this up kind of robustly. And so there's sort of two reasons why we felt really strongly about this. The first is that when an NTC, you know, normally people are having one or maybe multiple, hopefully multiple water controls on a plate. But you've typically got them, you know, they're only in a few parts of your plate. And if you're processing perhaps 96 samples in one go, if one NTC lights up, say in that bottom left corner, you often have to discard the whole plate because, as Nabil pointed out, you know, you don't really know where that signal is coming from. You don't know if that's an isolated incident or something systematic that's affected all 96 samples in your plate. And so normally, out of caution, you have to sort of discard the whole thing, or at least a large portion of it. And this, when you're, you know, needing to process many samples all the time, this is a big cost hit. It's a big time hit. Also, these standard layouts will miss sporadic small contamination events, say in the middle of your plate here. You know, your water controls will probably look fine in that case, but you've actually got a contamination event that you don't see. The real initial reason why we got into this was because we were doing a lot of cluster investigations, working with infection control groups at local hospitals or the Massachusetts Department of Public Health, where we were using this sequencing to rule in or rule out transmission events in small clusters. So these were two sort of nosocomial clusters in a local hospital. And so any contamination like this could have led us to make completely the wrong determination on whether or not these sequences were linked or not, which has, major ramifications on how people are understanding infection control practices, transmission risks, and all sorts of things. Obviously, and this has been alluded to multiple times already this morning or this afternoon, I guess, for you, this has really come to the fore in the last few days with Deltacron and, just what a dramatic effect contamination can have when it's not checked and caught early on. So the approach that we settled on for these in-well sample controls was we wanted to have something that was specific, that was unique, but that was also really easy to integrate with the ARTIC sequencing. We didn't want this to be a laborious process because we're already up against a big pressure to sequence a lot of samples. So what we decided to do was to have a unique sequence that was going to be specific to each sample that we were trying to tag, but then have a common flanking region of the primer sites that would amplify it. So it works a lot like an ARTIC, a regular ARTIC PCR amplicon. The core is 140 base pairs long of uniquely identifiable sequence. We have 96 of these different sequences. We typically work in 96 well plates, so that would allow us to add a single into each sample. And they're from, we decided to take an approach where we took these from genomes of uncommon archaea. So these were things that we wouldn't expect to see in our samples. And we confirmed bioinformatically that there was no homology with the human genome or with any common microbes, known pathogens that might lead us to misclassify these things. The way that we use these is that we, as I said, they're amplified by a single primer pair. So we mix this single primer pair with the ARTIC primer pools with both pool 1 and pool 2. And then we add a single SDSI per sample in our plate. So that's kind of shown in the graphic here. Normally we add SDSI 1 to the first well and so on and so forth, but it doesn't really matter so long as you know which spike in you're adding to which well, so you can retrieve that information at the end. So we add a single one of these SDSIs. I don't think I defined it before it was on a slide. Synthetic DNA spike in is what we call them, and SDSI for short. We add that at the cDNA step. So after you've got your RNA, you've done the first strand synthesis and made it into cDNA, right before that amplification step, we add these spike-ins in. And so it's pretty minimal in terms of per sample cost or time. You just add into your master mix, you add in one extra primer pair. We actually pre-mix these, so we don't have to do that step. And then you just add your, as you add your sample, you also add your spike in. And then we add those in and then we process the sample normally. Everything else remains the same about the method. So whatever your amplification method is, whatever your library construction method is, and then whatever your sequencing platform of choice is. And then at the end, when we retrieve our sequencing data, we do one extra step. So as Nabil said, you pull out your SARS-CoV-2 reads and assemble those genomes. We do one additional step, which is we map those sequencing reads to a FASTA file containing all our SDSI sequences, and then we count those mapped reads, and we end up with something like these heat maps. And so the one on the left is sort of what you would hope to see. That's a good run where sample 1 had spike in 1, and that's really the only thing that you see. Sample 2, you only see spike in 2, and so on and so forth. And what we're looking to pick up is cases where it doesn't look like that, either where one sample or perhaps multiple samples fall off the diagonal. We typically use a threshold of 95% of the expected spike in order to filter a sample. So anything where we see more than 5% of a contaminating spike in, we remove that sample. But also beyond the kind of per sample level, this can be useful in these patterns, and I'll get into this later. These patterns actually tell us a lot about where these failures are happening and where issues are arising. So we did a bunch of pretty intensive optimizations to make sure these worked well. We did a sort of serial titration to figure out how much we should be adding in. We were aiming to have sort of 3 to 5% of all of our sequencing reads be coming from this spike in order that we can robustly detect it and we have enough reads to say when we see a contaminant. but also so that it's not, eating up too much of your data that should be going to SARS-CoV-2 and, making things more expensive or making your detection sensitivity of the genome less. So we settled on 1 femtomolar initially. We've upped that now that we've kind of scaled this up, just to allow them to be a bit more stable for a longer period of time. We just sequenced the spike-ins by themselves. We checked, theoretically, we knew that they shouldn't hit to other species, nor should they misalign to each other. And that indeed seemed to be the case. They were very specific in, the first spike-in only mapped to the faster of the first spike-in and so on and so forth. We did a lot of kind of intentional contamination to figure out how quantitative versus qualitative this was. We did sort of mixings of, 100% of a spike-in versus 50%, 75%, 25%. They are sort of semi-quantitative, we think, although we would need to dial this in more. We use it routinely as just a qualitative flag at this point, and even as that, it's pretty powerful. We also, and really importantly, saw no impact on genome recovery or per amplicon coverage. So this is the plot on the right here, where you're seeing the black line is just with the ARTIC primers, and the blue line is with the ARTIC primers and the SDSI primers and a spike-in added. And we don't see any difference in our ability to recover a genome or like per amplicon, the kind of interaction effects of that primer set with any of the ARTIC primer sets. And the genomes, I'm not actually showing this, but the genomes were also identical. We don't see any, it's not introducing any spurious mutations or anything by mispriming of other sites. And we trimmed those SDSI primers off in the same way we trimmed the ARTIC primers off that Nabil already spoke about. So once we had this kind of set up, we did all these optimizations and we got it to a point where we were happy with the performance in a test set of clinical samples. And then we decided to slot it into our standard processing pipeline for ARTIC sequencing, which we kicked off kind of about this time last year. So we switched from metagenomic sequencing to amplicon sequencing with these spike-ins as a kind of key part of our step. And we also at the same time actually sent these to a collaborating lab, Ryan Chewy's lab at Jackson Labs up in Maine, not too far away from us. And they were also just switching to amplicon sequencing and were interested in having a similar control. And this actually allowed us to assess not only sort of how well these were working at scale, but also how robust they were to variations in the lab context and, slight modifications in protocols that we were doing versus what Ryan was doing. And so we put them into our pipeline, and then we sort of kept an eye on their performance over the next sort of 3 to 4 months. And so that gave us sort of through the winter and spring of 2021, about 6,700 data points of how well these were doing. And so what we saw was we kind of looked at this in 2 different ways. The first was we were interested in seeing, overall, how often are we hitting this 95% threshold? Is it really an anomaly when something falls below it? And therefore, is it kind of a good way to identify purity? So overall, we found that the percent of, so the measure on the y-axis of this top plot here is the percent of SDSI reads mapping to the expected SDSI. So it's everything that maps to one of these spike-ins is the denominator, and the numerator is what maps to your expected spike-in. And overall, they're pretty consistently above 95%. So they do maintain purity. We're not seeing kind of contamination of our spike-ins, which would obviously kind of distort things. They're pretty robust across the 2 sites at being above 90% purity. We didn't take out here any samples that were flagged as contaminants. So anything that falls below the line are ones that we would have pulled out and not submitted to GISAID or to NCBI. We also wanted to check that we were still not taking up too many of our sequencing reads with these SDSIs. We had sort of dialed these in to be between 3 and 5%. Across the 7,000 samples and across both sites, that was roughly what we saw. They were typically around 5% of all of the sequenced reads that we generated were mapping to a spike-in rather than to the SARS-CoV-2 genome. In a couple of cases, this was headed up towards 10%, but it was on average never more than 10%. So these are not taking up a large amount of your data, which means they're not adding much extra cost or impacting your performance. And we didn't do this specifically. We just sort of put them into our pipeline and kind of ran with it. But when we sort of took the data out at the end, we realized this was actually also covering quite a nice cross-section of information. We had data from a lot of different samples with different CT values from different states and from different lineages. Obviously, this was in the days when there was still a large diversity of SARS-CoV-2 lineages to sequence prior to Alpha, Delta, and Omicron kind of wiping the board with all the others. And so, you know, this was just sort of part of our routine sequencing surveillance that we dropped these into, but they were performing pretty well across all these different variables, different submitting labs sending us samples from different processing methods, different sample types, and all sorts of different things like that. This is sort of my last data slide, and this was the piece that we found really interesting from implementing all of this, and from having this additional level of resolution about what was happening in our sample plates as we process them. And so I mentioned that we use this method, this in-well control, to filter out specific samples that fall below our designated purity cutoff that we think might be a measure of contamination. But it also showed us a number of patterns across different plates that I think identify kind of key failure modes and key places to really pay attention to as you do this ARTIC protocol, and as you think about kind of scaling up your own sequencing. And these were highlighted by the patterns on these plots. So I think this one on the second one in from the left is sort of what we were mostly expecting to see, right? Like neighboring sample contamination, where you get sort of like the schematic I showed at the beginning, a sort of spillover in one part of the plate from one sample to another. And we saw a number of examples of that in all different parts of the plate. We also saw a bunch of other patterns. So this one on the far left was a case where 2 of these spike-ins were detected in almost every sample on a plate. And what we think probably happened there was a reagent contamination. So we think perhaps these spike-ins got into either our primers or one of our reagents or something, or onto a surface that they then got contaminated into something, and they were present in all of our samples. So this plate, we discarded the whole plate, and we also discarded all of our reagents and started fresh. This one second in from the right was actually the most surprising one to us, where when we took out this data at the end, we saw this perfect X shape across our heat map. And we think what this is I mentioned at the beginning that we put the primers and the spike-ins into both pool 1 and pool 2 of the ARTIC primers. And then obviously in your protocol, you do the PCR of these 2 pools, and then you recombine them before you make your libraries. And we think what happened is that one of those plates got inverted. And so when they got recombined, the samples were fully kind of flipped. And this is actually potentially a good spot to pay attention to when you do split the samples and recombine them. That it was just one case, but it was a good illustration to us of the fact that these things can happen. And this was sort of a similar idea, but on a smaller scale, that we saw this potential column mixing. So there are parts of our protocol that are manual, and we think this is a case where one column got put again when they were the plates were recombined after the ARTIC PCR, that maybe one column was recombined incorrectly. And so while these cases are the exception, many of these would not have been detected by just a water control. And beyond just picking out these samples that were contaminated, they also identified these sort of risk areas in the process. So that's really all I wanted to share. I think as a sort of summary or recap, I think that the main thing I wanted to really emphasize is that contamination happens, and it's not a bad thing. It is a bad thing for the data, but it doesn't say something bad about your process. I think at the beginning, there was a lot of kind of guilt around like the idea that contamination happens. It's just a fact of life. We do so much PCR amplification in this protocol. And so when and we saw in Deltacron and whatever, like it can happen, and it quite possibly will happen, but the best thing you can do is kind of get ahead of it and just put in multiple layers of control. So you're being really, really thorough and looking at this both on the lab side and on the bioinformatics side to try to pick out cases where you think this is a problem. And particularly if you have samples where you're making important inferences from. These in-sample controls we found to be a really great way to help with this. And they added resolution both to specific sample swaps, which was kind of what we set this up for, but also bigger QC questions about the plate and the process. We are still working on these, so I just wanted to share a couple of things that we're doing right now. We're trying to get an RNA version set up. So these, as I mentioned, go in at the cDNA stage right now, which means that we did that because we felt that was the most high-risk area because of the possibility of sort of highly amplified material. being more likely to spill over or contaminate other samples. But obviously an ideal thing would be to track right from the beginning, so you had a proper end-to-end tracking. So we have preliminary data on RNA, which works pretty reliably, and we're trying to kind of set that up to be a more systematic thing that we integrate into our sample processing. We're also trying to adapt it to other approaches. It was designed with the ARTIC platform in mind, and that's what we've been mainly using it for, but it's pretty, like, theoretically very versatile and very agnostic to other multiplex amplicon methods. Right now, what we're trying to do is integrate it with some of these tailed primer amplicon approaches that people are doing, so we don't have to do the library construction on the end. And we're also still optimizing storage and performance scale. So we've had requests from a few folks to share these spike-ins with them, and we're trying to get lyophilization stuff set up so they're a little bit more stable longer term. I just wanted to quickly finish with a big shout out in particular to the 3 graduate students, Matt, Kim, and Erica, who really led all of this, who sort of gave up 3 or 4 months of grad school to come into the lab and help us process samples through the first wave, and were the kind of key masterminds behind our switch from metagenomic sequencing to amplicon sequencing, and really making us all comfortable with our ability to detect and track contamination in amplicon sequencing, which was kind of key to us kind of making that switch, as well as obviously a lot of other people in the group who were really important in the project and in kind of our general SARS-CoV-2 sequencing that this method is kind of has supported and continued to support, as well as folks at MGH, Mass General Hospital, who were sort of in that infection control team that we were doing all of this with, and Ryan and his group at Jackson, who were our second validation site. So I think I've left time for questions. I hope I have, and I'm happy to answer them if anyone has any.
Brilliant, thank you so much. I think it's a really nice method. I really like the demonstration of the different types of lab problems that you can pick up, and we know that this happens, of course. And I liked what you said as well, which is that, you know, it's not a bad thing. It's a thing that happens. It's inevitable. The trick is, can you pick it up and can you mitigate it? So I think this is just a fantastically useful method. I think you answered most of the questions that I had, but I guess the key question for many people would be, if they want to try it, how would you recommend they go about trying it? Should they go and get these synthesized individually, or as you said, you were looking into sharing and lyophilization, but what would you recommend for people that want to get started?
Yeah, I think if people want to try it, so we got them synthesized, I think, from IDT. Are IDT a thing in the UK? I actually don't know.
Yeah.
Okay, cool. I'm not sure how internationally things are, but yeah, so we did ours from IDT. Like, we synthesized them as like very high purity, just because we didn't want to have any kind of, well, as minimal background as possible from the, like, a baseline from the SDSIs. Oh, that's the other reason we set a 95% cutoff. I didn't mention that. There is some kind of like below 1% level of kind of background between them, which is unavoidable, sort of similar to primers. But we synthesize them high purity from IDT. Anyone who will synthesize kind of 140 nucleotide sequences at like high purity would work. We started out just by testing a small set and dialing those in, and then ordering the larger set. All of the sequences are publicly available in our manuscript. So we published all of the sequences that we used, as well as the primer sequences and everything. So you're very welcome to take those. And then if folks are interested in testing out, we don't have like infinite quantities, but we do have a decent amount. So if anyone, we are like very willing to share these within what we can manage. We're not we're not IDT.
Careful what you offer.
Yeah, so but I'm certainly happy to help with troubleshooting or anything like that if people want to try that.
All right, that's fantastic. Okay, there was a question in the chat, which from Lays, which was, are they only compatible with ARTIC v3, or can you use them presumably with other versions of ARTIC and other? Yeah, absolutely. Yeah, you can use them with other versions of ARTIC and other versions of other primer sets as well. So we are using them with ARTIC v4
they were, yeah, I had ARTIC v3 on one of those slides. That was sort of what we were on at the time we designed it. But yeah, we're now using it on ARTIC v4. There's nothing about the primers or the SDSI sequences that makes them sort of specific. We designed them to have pretty general melting temperatures, GC contents that should be, you know, relatively compatible with other things. And are certainly compatible with the ARTIC primers and the sort of v4 and we imagine subsequent updates. So they're just a standard primer pair and this insert, so you can kind of pop them into whatever your primer scheme is.
Okay, so they're basically generic, and you know, they seem to amplify well with the ARTIC schemes around the TMs that those primers are. So that's really helpful. And you've got 96. Were you thinking? Have you gone beyond 96? You might have said.
We've thought about it. We haven't got we've got kind of sequences and sort of theoretical lines. We've not validated a set beyond 96. But it's definitely something that certainly we work with the genomics platform at the Broad who are doing this at sort of much higher scale. It's something that they were interested in, like having, you know, they do more 384 well plate processing than 96. So yeah,
I could definitely imagine 384 could be helpful. And a follow-up question was is there an already available bioinformatics pipeline to perform the analysis that you showed?
That's a great question, actually. There should be. I believe it's on the, we have all of our pipelines are available under something called Viral NGS on DocStore. I believe the spike-in workflow that we use is part of that package. It's from the Broad Institute under Viral NGS. I will double check that tonight though.
Okay.
Yeah, I'm pretty sure it's publicly available.
All right, brilliant. Please, any more questions? We've got a few more minutes for questions. So if you have any questions, do pop them in the chat or put your hand up or whatever you like. And so another question I had, and you answered that, was you know, why use it at the cDNA stage? Why not use it at the pre-extraction stage or the, you know, the RNA stage? And you answered that. But if you had RNA spike-ins available as well as DNA spike-ins, would you envisage using both of those at different stages to look for kind of different points of contamination, i.e., you know, kind of a very early sample kind of mix-ups versus downstream errors? Or would you just anticipate you would just use the RNA ones if they were available? Yeah,
that's a good idea, actually. We were anticipating just using one and just trying to get it in as early as we could, because they do have the same primer sequence right now. So we reverse transcribe. We actually synthesized and I think that's in the paper, the sequences that we have in the paper, we synthesized them as DNA, but with a T7 promoter on them. So we could just transcribe them ourselves into RNA and test that out. So they, as a consequence, they're amplified current same primer sites. If you were to switch that out and we design an RNA and a DNA set with two slightly different primer sites, or at least a larger set of unique sequences and unique inserts as well, you could definitely do it that way and have sort of tiered controls.
Okay, yeah, no, that's very interesting. And in terms of what is the most common failure mode that you saw, you know, during your work? What was the kind of most common thing that would go wrong that your method would detect in the lab?
For the most part, it was picking up like these one-off little spillovers, these neighbor contaminations, as we called them, and it was pulling out. We were filtering out maybe between normally under 5 samples in an average run. So it's not something that was happening all the time ubiquitously, but it was something that was, you know, consistently. There was the sort of reliably, there was the odd one or two where there was at least enough of a concern that we would sort of re-sequence and re-prep that sample. The bigger issues were less common, but were nonetheless interesting. Yeah. Okay. So this is kind of like almost like single pipetting errors and things like that, or like small spillovers and stuff like that. Yeah. Yeah. Okay. Brilliant. All right. A couple more questions before we finish. Have these spike-ins been tested on wastewater samples? Oh yeah, that's a good segue into the next talk. We've actually just started that. So we were primarily doing clinical samples for most of the time. We've been working mostly with public health and with hospitals on patient samples, but we have recently started some more wastewater work. So we're trying to integrate these with the wastewater. A priori, I mean, there's no reason why it shouldn't, aside from, I think the thing we're interested in is just, what that diversity of material in wastewater does, and if that has any impact on our on misclassification. It shouldn't, because these were designed to be very unusual sequences. But that's we're looking into that right now.
Yeah, that makes perfect sense to me. Okay, and could you add the link to
I will, yes,
to the chat? That would be great. I put it on Twitter already, but if you could add that, that'd be amazing.
I will.
Okay, well, I think that's all the questions, and that's time. So thank you so much for joining us. It's really appreciated, and it's really interesting.